Yes,
Did
sir.
Meenakshi remember? Yes. Okay. I think we completed till Cleotropy, right? In the last class. You were there, Reem, in the last class?
Yes, sir. I was there.
What did we do? We did combination, crossing over, right?
Yeah.
Linkage. I think we started from where? Chromosomes. Chromosomal theory of inheritance. We start from, right? Yeah. So let's begin with a quick recap. Reem, would you like to do the honors? Just
Sir, I did last week, right?
you did last week.
Yeah.
What? So like this we did last week, right?
Yeah.
You were there in the class.
Yeah.
Yeah. So just give me a brief recap.
Sir, I'm not prepared.
Okay, just I'll help you. I'll help you. Let's begin with what did we discuss? So the first thing we discussed was chromosomal theory of inheritance.
Yeah, it was given by
That
two
is
sign.
Yeah, that is the point of me taking this, because I do it on purpose. So even if you're not prepared, you will realize that you should have been prepared. At least before coming to class, you could have revised. So it will just give you a message, and it's not a test. So whatever you will forget, I will add on to it. Okay. So relax.
Okay,
Let's
sir.
start. Yeah. Okay.
This theory was given by two scientists, Sutton and Boveri.
Yeah.
They did it individually, but then
Yeah.
they did it together, but in different places.
Yeah, which means not together.
Yeah.
They at the same time, but independently they figure out this thing.
Yeah.
And what about these three scientists? Why were they important?
They made that Mendel's work
As laws.
Yeah, like they made it.
They give the work the status of laws, the three laws that we have studied. They also independently rediscovered Mendel's work after a very long time.
Yeah, Mendel's work was not recognized, but they did something to make it recognized because they were actually biologists.
Yes, correct. Yeah. Now we also discussed chromosomes. Tell me, what are homologous chromosomes and everything?
Chromosomes have two parts. One is short arm and long arm. Short arm is known as the P arm, and the long arm is known as the Q arm. And they have this thing on the middle. And it's
called centromere.
Yeah, it is called centromere.
And this is where
when they replicate, the chromatids are known as sister chromatids, and different chromatids are known as non-sister chromatids.
What do you mean by different and same?
Like the individual.
So sister chromatids are the exact copies of each other. They are formed from the same chromosome, right?
Yeah.
Whereas non-sister chromatids are the chromatids of two homologous chromosomes.
Yeah.
Different, but yet homologous. They has to be the same chromosome from two different parents. Okay?
Yeah.
Yes. Good. Continue. We also studied about locus. What is locus? Gene A, gene B.
The
locus.
Location. Locus is the location.
Yeah. So why did you say you are not prepared? You seem to be really well prepared. Okay. And what was chromosomal theory of inheritance? What did the theory say? Yeah.
Sir, I don't know. I'm not prepared.
Yeah. So it says that, just like Mendel says, that there are factors, or we call it alleles, which pair or separate, right? So when they pair or when they separate, they are actually doing it with the chromosome. So pairing and separation of a pair of chromosome leads to segregation of a pair of genes that they carry. Yes. So when you pair chromosomes, you are pairing genes. When you are separating chromosomes, you are segregating genes. So this is known as chromosomal theory of inheritance, simply. So it is just, in another words, an advanced version of Mendel's law. Okay. Then
sir.
we went on to do linkage. You remember what was linkage? What is linkage of genes? It is an
When two
exception.
different alleles they come together.
No, it has not. It is not related to alleles. It is, remember when Mendel said that when there are two genes, it is the exception for law of independent assortment, right?
Yeah.
Linkage of genes is an exception to law of independent assortment, which is the third law. So here, a scientist Morgan discovered that there are certain genes which do not segregate independent of each other. For example, he did experiments in Drosophila and he figured out that yellow bodied the gene for body color and the gene for eye color. A yellow body Drosophila most of the times also has white eyes, and a brown bodied mostly has red eyes. And these two genes are linked to each other, which means when you make a cross in the F2, they do not follow the 9:3:3:1 ratio. This should be the ratio that normally two genes must follow according to Mendel's law of independent assortment, the dihybrid cross. Remember, everyone?
3:1.
9:3:3:1, right? 3:1 is in the for the one gene interaction. Okay.
Okay.
We also have Saifullah. Saifullah, how are you? Good evening.
Yes, sir. Good.
Yeah. We're just I'm giving a recap. Me and Reem. So linkage is where two genes are linked together so that they do not segregate independently of each other all the time. Okay. And recombination is when the linkage is more, you will not form many different recombinants, right? Saifullah, would you like to help Saifullah in this recap? Will you explain from recombination onwards?
I will try, sir.
Yes, please. I'll be there. We'll do it together. But you lead, I'll follow.
Like,
What is recombination and how is it associated with linkage? What happens if the linkage is more, the recombination is
less.
less, which means more the genes are tightly linked together, less are the chances of they going independent of each other in the gametes and forming different variations. So recombination is rearrangement of DNA sequences, right? See,
Yes,
these
sir.
parts are a little difficult to understand. So that's why I am again asking you all to please revise. And I think you have received, Saifullah, my WhatsApp message on the group.
Yes, sir.
Okay, very good. I told you how does recombination happens. It happens through crossing over of chromosomes. This image, this figure, we spend a lot of time on this. Saifullah, would you like to explain this? What is happening here? What are these blue and red dancing things? Why are they doing
Yes.
bhangra together?
Like, during cell division, there is a method called crossing over in which a part of a gene of two non-sister chromatids are interchanged,
Yes.
which leads to other mutations and
No, not mutations here. There's no point of mutation has yet not come to the mutation is a different thing, which leads to recombination.
yes,
Mutation
exchange
is
of
change.
genes like that.
Yes, mutation is the change in the genetic code. No change is happening here. It is just exchange happening. There's a difference between change and exchange, right?
Yes, sir.
You had a 10 rupee note. I have a 10 rupee note. We exchanged it. You had a 10 rupee note. I asked you to make it a 20 rupee note and then change it. That is called a change. You understand?
Yes, sir.
The note changed. It is not a 10 rupee note anymore. Okay, continue. Then what is happening in the third diagram? First, second is exchange. Then in third, what is happening?
They are separated after crossing over. They'll be
Okay.
going in different cells or gametes.
Yes, yes. So how many chromatids will be recombinant chromatids and how many chromatids will be non-recombinant in this case? chromatid 1 and 4 here in this picture. 2 are
recombinant.
Non-recombinant, parental types. Right? Non-recombinants are also known as parental type. They are just like the parents, no?
Yes, sir.
Do you realize? This one, capital T, capital Y, capital R is just like the parents, right? And small t,
Yes, sir.
small y, small r is like the parent. Whereas these two, 2 and 3, they are recombinant chromatids. They are different from the parents. They give rise to variation. Yes?
Yes, sir.
Yeah. And then we discussed that linkage is inversely proportional to recombination. This is the formula to calculate frequency of recombination. Everyone has written this formula?
Yes, sir.
And then we did some questions on the basis of it. We figured out 10% was the recombination rate. Yes. And towards the end, we discussed two more concepts, polygenic inheritance and Cleotropy. These two are non-Mendelian forms of inheritance. So they do not. What was polygenic inheritance? Mamona. Are you there, Mamona? Mamona, can you speak? Yes or no? Okay. We have Madhiha joining in. Madhiha, how are you? Good evening.
Good evening, sir. Sir, I actually just joined to tell that I have an exam tomorrow, my last exam physics. So I have to study right now. I will be joining classes from tomorrow.
Okay, so you will not be continuing today?
No, sir, not today because I have physics exam tomorrow, so I have to complete the revision.
Okay, so that's up to you. But please do get your hands on the link and look at the classes, okay?
Yes,
Because
sir,
we are
I'll get
studying
the links
genetics.
and
Are you
everything.
doing this, right?
Yes, sir. Okay.
Okay, very well, fine. Mamuna has some
Thank you,
issues
sir.
with her mic. She's not able to speak up as she's writing. She's present there. Okay, let me go to Minashi. Minashi, are you there?
Yes, sir.
Yes, what was polygenic inheritance?
It means a single character is controlled by 3 or more genes.
Yes, perfect. Polygenes. And please explain with one example that we have discussed.
Example in color.
And what did we do here? What is on the screen? Can you see the screen? What was this A, B, C, D? We assume that, let's assume there are 4 genes which control. Then what will be the darkest skin color? This combination, right? Everything capital, everything dominant and expressing. And everything recessive or not expressing will be the fair skin, the recessive one. And why there is skin color? Because of what biological factor? Skin color arises due to a pigment, right?
Yes.
A pigment protein. What is that pigment protein called?
Melanin.
Yes, it's called melanin. And it is also influenced by environment, right?
Yes.
So more exposed to sunlight, darker the skin, less exposed, lighter skin color, right?
Yes, sir.
So polygenic inheritance or the polygenic genes can also be affected by the environment along with the genetics. So this is the genetics and this is the environmental factor that affect human skin color, correct? Very good. And also, what was Cleotropy? This you remember, Reem, Cleotropy, the last part.
Yes, in this, a single locus affects two or more traits.
A single gene, just like the opposite. There is one gene,
Yeah,
but it's influencing multiple phenotypic traits, right? Expressions. Perfect. And we discussed the example, one example called phenylketonuria. Yes. Can you tell me what was this phenylketonuria?
It is caused by mutation
Mhm.
that codes for enzyme.
Mhm.
And then
Which enzyme? It is a mutation which is caused in a single gene, remember? And I told you, normally we will believe that if there's a single gene that is affected, there has to be a single trait that is affected, right?
Yeah.
According to Mendel's rules. But here it's not the case. The enzyme codes, the gene codes for the enzyme phenylalanine hydroxylase. And this single gene mutation affects a lot of systems. So if it is doing mental retardation, it is affecting which system? Nervous system? Yes?
Yeah.
It's also affecting hair pigmentation and skin pigmentation. So it is also affecting the the epidermal layer, right? The skin.
Yeah.
And in this case, it is affecting the hair. So all these three are different systems, right? Different.
Yeah, but they're caused by one gene.
They're caused by one gene. So that is what it means by a pleiotropic gene expression. Is it clear?
Yes, sir.
Very well. So now we will start with, let me add one different page. Let's start. Today we'll be starting with, it's still a part of like chapter 5, but I just want to start a new notebook because it's very cluttered there. So here we'll be starting with genetics of sex determination. Sex determination in organisms, okay? We already know about sex determination in humans. Yes or no? So
Yes, sir.
first tell me, how is, what is sex determination of humans? Sex determination in, just 1 minute. Is the screen still visible to all of you?
Yes, sir.
Yeah. So sex determination in humans. Tell me, what do you know about sex determination in humans? Reem, we have discussed this in chapter 3.
It determines the sex of the offspring
Yeah, but how it is done in humans?
by chromosomes.
Yes, it is genetic in chromosomes. And what are the sex
Yeah.
chromosomes in human? There can either be a X chromosome
X, Y.
and is there any third one?
I don't think so.
Yeah, there are 2. And the combination is, if there is XX,
It's a female.
it is a female, right?
Yeah.
So females are heterozygous or heterogamitic for sex determination, and males are, sorry, homozygous. Females are homozygous for the sex determination, or homogamitic, and males are heterogamitic, correct? Having 2 different chromosomes. So when this female will go on to make gametes, she will make 2 different gametes. So sorry, she will make both the same gametes. Each gamete will have X or X, right? Is it
Yes,
understandable?
sir.
So
Yeah.
this is called homogamatic condition. All the gametes are same, but in males, there will be 2 different types of gametes. One gamete containing X chromosome and the other gamete containing Y chromosome. So this concept is called heterogamity. We have discussed it earlier also, but just to elaborate it here in this chapter. Is this clear to all of you in humans?
Yes, sir.
Perfect. Now write down, in humans, there is male heterogamity. So if the question comes, this is a this has been a question in boards. What is male heterogamity? What is male heterogamity? So you can write down, the in case of human males, they produce two different types of gametes. Two different types of gametes containing two different sex chromosomes. Containing two different sex chromosomes. In bracket, you can write X and Y. This phenomena is known as heterogamity. Okay. Is it clear?
Yes,
In
sir.
some,
Yes, sir.
yes, in some species of organisms, there is female heterogamity also. So write down. In many birds, in other organisms like birds. So write down this question as well. What is female heterogamity? Which organisms have it? So we can write down. In organisms like birds, females have a pair of different sex chromosomes, which is the case in human males. Different sex chromosomes. In the bracket, write down Z in W. This is called ZW system. ZW system or ZW system. Humans are known as XY system. Okay. So for male heterogamity, the examples are humans and fruit fly. Fruit fly is known as Drosophila, scientific name. So these two have XY system, where males have XY. In birds, females have a pair of different sex chromosomes, which is called ZW. And they form two different gametes. One containing Z and one containing W, correct? This is female heterogamity. In this, you can write males have two similar chromosomes, sex chromosomes. Two similar sex chromosomes, which is Z and Z. So this W is different in females. How you can remember it? W for woman. Yes. So W is only present in females.
Yes, sir.
So this is a different system. This is where female heterogamity, and both these questions have come multiple times in your board. So you remember this. Okay. So we have to study sex determination in certain organisms, in which they always ask about human, Drosophila, which are same. Human and fruit fly have this XY system, and the sex is decided on the basis of X and Y. If X and Y is present, heterozygous, then it is a male. If it is homozygous, it is a female. For birds, they ask for chicken. Example here is chicken. Okay. For that, you know that if it is Z and Z, which is homozygous, it is male, opposite. And if it is ZW, it is female. W is for woman. This is how you can remember. Now, another important one is, write down, sex determination in honeybees. This is a favorite topic of entrance examinations in honeybees. Now, in honeybee, it is slightly different from both XY and ZW system. Okay. Do you know, first of all, honeybees are social animals? Honeybee are social animals, right? What do we mean by social animals? Any idea? Anyone has seen how do honeybees live? Where do they live? Are honeybees living like cockroaches or mosquitoes, like independent?
In their beehives.
They always live in a colony, like ants, termites, right? That's why ants, termites, and honeybees, all of them are called social animals. They live in groups. And they are called social because in this group, there is a division of labor. Right? So honeybee lives in beehives. And these beehives, there they have a division of labor. What do we mean by division of labor? Work is divided. Some specific type of honeybees will do specific type of work. Do you know about it? Tell me, yes or no? And no one knows about honeybees, so let me tell you. Honeybees are interesting because in this beehive, there is only one single queen bee. You know that?
Yes, sir.
And this queen bee, she is a female. She is the only female fertile bee. All eggs which are given in the beehive are laid by this. So all eggs are laid by queen bee. This is the only female fertile which is capable of reproduction. Is it clear? Apart from a queen bee, there are worker bees. You know worker bees? So queen bee is a female. Worker bees, are they male or female?
Male.
They are females. All worker bees are females, but they are sterile. Sterile females incapable of reproducing. Is it clear?
Yes, sir.
Yes, sir.
Yes. Yeah. So the only bee that is reproductively active is the queen bee, right? Worker bees, then how are they sterile? Why are all the worker bees sterile? You know, there is a reason, interesting reason behind it. There is something called royal jelly. It's a very nutritive thing. Royal jelly is only fed to the queen bee, which makes queen bee bigger. She is bigger in size. Okay. So this is one way to identify it. She is big in size, bigger than all other bees. And queen bee secretes a pheromone. You know what is a pheromone? People, pheromone. Pheromone. It's a chemical that is produced from one animal's body. It's like a scent or smell, which attracts or gives signals to other organisms. Have you ever seen ants? Moving in a very, suppose there is a stone kept here. Have you seen ants moving like this?
Yes, sir.
So how do they follow? How do they know they have to follow each other? If I take a finger and I disrupt this from the beginning, then what will happen? You all must have done it. As kids, we used to see ants. Have you not done it? Have you seen ants around you?
I guess they're attracted to that smell.
So for some time, they are wandering, right? They become haywire. But again, after some time, you know, if one leads like this, they will change a little bit, but they will again form a line. Yeah, they always form a line because the ant which is going in front keeps secreting pheromone and leaving a trail. It's just like footprint. So in ice, if humans are moving, you will see one footprint and you will follow the, you will just put your feet on the same footprints, right? It's just like following each other's footprint. This is also because of pheromones. Is it clear, everyone?
Yes.
So this queen bee secretes some pheromones. Now this time, this pheromone keeps all these worker bees in the hive sterile. So they're sterile because queen bee secretes something which keeps all of them sterile, but they are female. Then the third kind of bees are the drones. Their only thing is to protect the hive, to look for any potential threat, to go and forage around and look for where the food is present, then come back and give signal to the worker bees that, see, I found food there. So all the worker bees, not all, but some worker bees will fly and go and collect the nectar and come back. So drones are basically males. Okay. And they are fertile. They produce sperms. But all the drones actually can reproduce with whom in the nest, in the hive? Queen bee.
Queen bee.
Is it clear? So this is the division of labor. Queen bee secretes, maintains, keep the hives as a unit. She is the leader. She is the one who keeps the hive as a hive. She is the one who keeps worker bees sterile and keeps them as workers so that they keep cleaning, making, expanding the hive, collecting the nectar, making the honey and all those things. Basically, you know what is a honey? It's gross to hear, but it's very nutritive. Honey is bee vomit. Bees, they suck on nectar, then they keep the nectar in their stomach, which is like half digested. Then they come back to the nest and they regurgitate all the nectar which they have kept in their stomach back in those cells, hive cells. You know, a beehive is like hexagonal in structure like this. Yes or no? You understand what I'm drawing, people? And inside is honey filled, right? So honey is basically bee are vomiting. It's bee vomit. but it's very, very nutritious. Drones are fertile males which can only have which can reproduce with the queen bee, or they do not reproduce at all. Okay, is it clear? Now here how is the sex determination happening? How is the sex determined? So basically all these workers and drones are kids of queen bee, if I can say like that. Can I say like that? Because only queen bee is giving eggs, right? So all of them are basically queen bee's kids. Yes?
Yes.
Yeah. Now, write down, in bees, the sex determination is based on, in bees, the sex determination is based on the number of sets, of chromosome, the number of sets of chromosomes present in an individual. Present in an individual. Okay. Next line, write down. All offsprings, write it like this. All offsprings produced through sexual reproduction. Through sexual reproduction, which means, what does sexual reproduction means? In bracket, write down, union of sperm and egg. So all the offspring which are produced through sexual reproduction, union of sperm and eggs, develop into a diploid female. Is it clear?
Yes, sir.
Females are always diploid. So their sex determination is based on the number of chromosome sets they have. If it is a diploid set, then it is a female. And there are two types of females, either queen or workers. Both are produced the same. So can you now imagine that at one point of time, the queen bee was just a egg, a common egg among all the eggs, right? Worker eggs and queen eggs are the same because they both are diploid sets, right? Then what makes one queen and others stay workers? So what happens is, before the queen is about to die, she or the other females decide who will be the next queen. And now that is not done democratically with the vote. One out of all the eggs, which are female eggs, is chosen and is fed by the royal jelly. So when the larva come out of it, that larva feeds on royal jelly, a very nutritive medium which other worker bees do not get. And the one who is feeding on royal jelly will be just one and will develop into a queen female bee, which is diploid and fertile. Is it clear? Very interesting social setup, no?
Yes, sir.
Yes. So females are diploid, which means, okay, first write down the second scenario, write down. An unfertilized egg. An unfertilized egg which means just the egg. Always develops into a male. Develops into a male known as drones. So if it is a haploid set, then it will develop into a male. Correct? They are drones. So all the males are haploid. Their whole body is haploid. Not only the gametes that they make, but their whole body is haploid. Is it clear, everyone?
Yes, sir.
So this means that males have half the number of chromosomes as females of the same species. Now this is very interesting because when it was discovered, it was mind-boggling because see, males have just in case of humans and other organisms, I told you, right, that chromosome number should match, it should be the same. Here it is exactly half. So in this case, what do you think? Males, if they have to produce gametes, will they produce gametes by meiosis or mitosis? In bees, a drone, a male, is fertile, right? I told you in the beginning. If they're fertile, they make gametes. So drones makes gametes through meiosis or mitosis?
Mitosis.
Exactly. They are the interesting organisms. We make gametes through meiosis. And in fact, meiosis only happens in our body to make gametes and nothing else. In a male honeybee, in a drone honeybee, meiosis does not happen at all because there is no need of meiosis. They are already haploid. So write down, drones make sperms through mitosis. And this is a neat question. The question was, gametes are formed by the process of dash in drone honeybees. So if you just see gametes, the option will be mitosis, meiosis, both, none. You will choose meiosis and it's wrong. Drones make sperms through mitosis. In fact, there is no meiosis happening in drones. Write down. No meiosis happens in male honeybees. Interesting, right?
Yes, sir.
So let's do this. Let's start with one interesting thing. So if there is a female to begin with, let's start with the female. So there is a female honeybee. And if she is reproducing, she is the queen, right? Because queen secretes pheromones, which keeps, so the moment queen is about to die or she dies, what will happen is all the females will, the ban will lifted. So they can, when they get out of the effect of pheromones, they can again start becoming fertile. And that is a problematic thing because now when they are fertile, they will like to reproduce and lay eggs, right? Yes or no? And there will be lots of eggs, no space to keep, and no one will be working now because everyone is in the sexual drive. Remember sexual drive, chapter 3, Reem, others, Saifullah.
Yes, sir.
Yes, sir.
So
Yeah.
if everyone is busy finding a mate, finding a partner to mate with, then who's working for the hive? No one. Then the whole system, discipline of hive will go out. That's why there is a queen bee to keep a check on everything. What a beautiful system, right? Yes or no? So let's say there is a female honeybee to begin with, and there is 32 chromosomes in a female honeybee. 32 is the 2n number, okay? 2n is equal to 32. So there is 32 chromosomes in honeybees. How many are there in humans? 46. 2n number is 46, right? Their 2n number is 32. So this female will form gametes Okay. And the gametes will be formed through mitosis or meiosis? Tell me. Female will form gametes through what? Mitosis or meiosis?
Meiosis.
Yes, meiosis. So she is diploid. Only diploid cells can undergo meiosis. Remember I told you this? And after meiosis, she will form gametes, let's say, which is, so the gametes will have 16 chromosomes, right? This is the N number. Yes or no? Now, if this gamete is fused with another gamete, which is another N haploid, then this will give rise to another female, right? Yes, having 32, 2N. But if this gametes remains unfertilized, then it will give rise to 16, which is N, and which is a male animal. Is it clear, everyone?
Yes, sir.
So this process in which there is no fusion of gametes taking place is known as parthenogenesis. Remember plant system we studied, plant reproduction, parthenogenesis, development of an embryo without fertilization. Are you remembering something?
Yes, sir.
People? Yes. So write down parthenogenesis is development of an embryo without fertilization. Okay. So this is an interesting one to begin with. Now, if we are starting with male, this is what we are starting with female. Let's say if we are starting with a male, so the male will have what will be the chromosome number of the male?
16.
Quickly. 16, and this is haploid, right? Then it will form gametes, and gametes will be formed by mitosis. And when the gamete will be formed, there will be, the gamete will also have 16, right? And then it will fuse with another gamete of the female and will give rise to female, right? Do you understand? So male honeybees always give rise to females. If there are males, there will be more females which will take birth. They can never give rise to a male, right? Male is an unfertilized egg. So only female honeybees unfertilized eggs becomes male. Is it clear, everyone, for honeybees?
Yes, sir.
Please
Yes,
make these, please make all of this and let me know when you're done. So here it's simple. Haploid, unfertilized, parthenogenesis means male. Diploid, union of sperm and ovum means female. And females are of two types, queen and workers. Only queen is fertile, workers are suppressed by the queen through a pheromone. So they never reproduce. Make it and let me know when you're done. And this kind of sex determination system in honeybees, I'm writing it, it's known as haplodiploidy sex determination. Okay, everyone? Haplodiploidy sex determination. Now there is an interesting question that has been asked. They will just trick you on this concept. Everyone done? Then I'll ask the questions.
Done, sir.
Done, Reem? Okay, good. Minashi is done, right?
Done, sir.
Done. Saifullah is done. Reem. Mamun is also. Okay, very good. Now tell me, every male in the beehive, does he had a father? Do the male bees had a father? No. You understand? Any male bee did not had a father. It only came from an unfertilized egg cell from the mother, right? Is it clear, everyone?
Yes, sir.
So male bees do not have a father. They never have a biological father. So they just have mothers. Do female bees have fathers? Reem. Do female bees have fathers?
No.
Yes, of course. Female bees are diploid. They are formed from the union of a male sperm and a female ovum. You just wrote it above. Big. Union of sperm and egg develops into female. Females has to have a father. Where does the sperm came from otherwise? Yes?
Father, yeah.
So all female bees have fathers, but all male bees have only mothers, not fathers. Write down this statement. If you know this, it will be just 10-second question. And this has come as a question. Male bees do not have fathers. Okay. Female bees have fathers and mothers, both. Male bees only have fathers. Can male bees have grandfathers? Now this answer will be given by Reem because she gave a wrong answer last time. Reem, this is your redemption question. Do male bees have grandfathers?
Yes.
Why?
Because they have mothers.
Yes, and the mothers came from a father and a mother, right?
Yeah.
Yeah. So write down, male bees do not have fathers but have grandfathers. Do female bees had grandfathers? Minashi.
Yes.
Yes?
Yeah.
But only on the maternal side, right? Female bees had a grandfather because their mother had a father, but their fathers do not had a father. So they only had grandfathers on the maternal side, not on the paternal side. Is it clear?
Yes, sir.
Yes, sir.
Now let's come to sons and daughters. Will males have sons? Any male bee, will it ever have a son?
No.
No.
Male bees cannot have sons. They can only have daughters. Okay. What about female bees? Can they have sons? Of course. Their unfertilized egg becomes sons. And can they have daughters? Yes. They can have both sons and daughters. Yes? These are all the things that are asked. So, it's an interesting thing. You just don't have to know that one line from NCERT that haploid are males, diploid are females. All these things are also important. Okay, sons is clear, son and daughters. Females can have both sons and daughters. Now, can males have grandsons? Can males have grandsons?
No.
Think again. Males can have daughters and daughters can give rise to sons.
Yes.
So males can have grandsons, right?
Yes.
Can males have granddaughters? Yes.
Yes.
Yes. Keep writing. Males can have grandsons and granddaughters. What about females? Can females have grandsons? Of course. Right? Of course they can have grandsons. And they can also have granddaughters. So everything is clear about haplodiploidy sex determination in honeybees. And you understand why it is very interesting, because lots of questions can be made in these concepts. Any doubt, people?
No, sir.
No? Others?
No doubt, sir.
No doubts? Okay. So this was, I think, almost everything about sex determination. They have not, then they do not ask about environmental factors related, but you should know that for some organisms, even the temperature decides the sex of the baby. Like in case of many reptiles, if their eggs are incubated at higher temperatures versus lower temperatures, they develop into different sexes. You know that? So I'm giving this bit, it's interesting, as a homework. Do find out, write down your homework. Apart from genetics and haplodiploidy, sex determination, what are other types of sex determination? When you will Google this or try to find out this, you will find that there is one called temperature dependent. So I'm already telling you, temperature dependent. You have to figure out examples of it. Okay, examples and mechanism. Will you do this? Interesting.
Yes, sir.
Nature is interesting. So this is your homework. Note down. We will discuss this tomorrow after the test. Now, let's go to one more concept. It is an important concept, which is known as mutation. You know what is mutation, people? You have studied mutation in your junior classes, or at least the definition of it. What is mutation? Tell me, mutation, people.
Change in the DNA sequence.
Yes, it's an sudden and abrupt change in the, or alteration in the DNA sequence. So write down. Mutation is sudden, random alterations or changes in the DNA sequence of an organism. which leads to changes in the phenotype as well, correct? If there is DNA changing, which means the genotype is changing, then there has to be changes in phenotype. Correct? Now, if it is a change in DNA, then can mutation be inherited is the question. Can it pass on from one generation to another? Tell me yes or no. What do you think?
Can you repeat the question again?
The question is, can mutations that happen in an organism during one's life, do you think they are inheritable? Then can they be passed on to the next generation? So they are acquired. Mutations are acquired. They are changes that you did not, like it happened suddenly and randomly in the DNA of an organism, which leads to the changes in the phenotype of that organism as well. But do you think it can also be inherited? Yes or no? What do you think? What is your thought process? Even if it is wrong, it's okay. Like bring something to the table so that we can discuss. What do you think, Saifullah?
I think yes.
Yes, you think yes. And why do you think yes? What is your rationale behind thinking yes?
There be changes, and changes will be permanent, right? So if they will be reproducing, then the DNA which got changed will also be acquired by the offspring.
Yes, but you are correct, but you forgot one thing. Offsprings are only formed by the fusion of sperms and ovum. Yes.
Yes.
If there is mutation anywhere in the body which does not reaches the gametes, if there is a mutation in the gametes, only then it can be inherited. Do you understand?
Okay, sir.
Yes. So yes, mutations can be inherited if it takes place in the gametes. So write down, mutations are inheritable. Mutations are inheritable only when it takes place in the gametes or are carried by the gametes. Okay, is it clear? Which means they are sudden, random, inheritable changes in the DNA. There are many acquired changes in our life which do not get inherited. For example, if someone is a very good athlete, has very strong legs because he's a runner, then it's not like always necessary that the kids will have stronger legs too, right? Because that is an acquired character through training. Same is like intellectuality, intelligence, IQ. All these things are not inheritable. You understand?
Yes, sir.
Yeah. Now, let me ask you a question. If I'm doing an experiment with mice, okay? You know that mice has tail? Yeah. Yes or no? Mice have tail? Mouse? So if I cut the tail of a mouse and I cut the tail of two mouse, one male and female, and I make two tailless mice reproduce, then will the progenies be tailless as well? What do you think, Minashi?
No.
Why? Parents were tailless. Then why aren't babies tailless? What is the problem? Mamuna? Oh, Mamuna is not having some issues with the mic. Okay. Mamuna, please get your mic fixed if you can, because you are missing on some discussions then. Saifullah, what do you think? Two tailless mice, I cut the tails of both and made them reproduce. So they are tailless. Will their progenies be tailless?
No, since it does not affect the gametes.
Yes, because it is a physical alteration, right? It is equivalent to a male, a human, whose hands got cut, let's say, or leg got chopped off in an accident. So because of any reason, if a human does not have a hand or a leg, it does not mean that the babies are going to born handless or legless, right? Because the gene is there in the gamete. He was born with a hand, the human. The mice was also born with a tail. I just cut it physically. So this means that certain characters which are acquired or happen to an organism, if they do not happen at the level of gametes, they are not inheritable. Mutations also, if they happen at the level of gametes or if they are carried by the gametes, they are inheritable. Okay. So two tailless mice. Sorry. So if there are two tailless mice which are crossed together, let's say, their progenies still will be tailed, right? Tail cut, tail cut, tail present, right? These are the parents. This is the F1. Is it clear, everyone?
Yes, sir.
Now, I'm telling you, is it possible that two mice having tail, now tail is not cut, they have tail, but can still produce a tailless progeny? Question is, write down the question. This question has been asked in. Can two tailed mice give rise to a tailless progeny? If yes, under what conditions it is possible? Can two tailed mice reproduce to give rise to a tailless progeny? If yes, how is this possible? What do you think? Can you think of any? Like, you just have to be a little careful and innovative. You will get the answer. Who would like to try? Saifullah, just tell me, do you think it is possible or it's never not possible?
Can you say again?
Two tailed mice, two mice having tail, can they produce a tailless offsprings? Is it possible? If yes, how is it possible?
Maybe no.
No, so it's never possible. What do you think, Minashi?
Yes.
Yes. Why do you
Yes.
think it's possible? How?
Maybe some disorder, genetic disorder. Something.
Yeah, or it can have mutation, right?
Yeah.
Let me tell you how. There is a mouse which has a tail, but it has a mutation M plus on one chromosome, one set. On other chromosome, there is no mutation. So it is wild type. Okay. Mutation over wild type. This is a heterozygous condition. on one chromosome, either from father or from mother, the gene for tail development is faulty. It has a mutation, which is M+, mutation plus. On the other hand, it is normal, wild type. Is it clear? If it is crossed with another mouse, so because one was okay, this mice had tail. The other mice also has tail, and that is also heterozygous for that mutation. When they produce, there's a possibility that some of the kids can have mutation over mutation, both the faulty copies. In this case, there will be no tail. But the parents had tail. Do you understand, everyone?
Yes, sir.
Okay. So this is one possibility through which, yes, it can happen through this possibility. So mutation over mutation. Okay. So no tail. Make sense? Now, what are the factors that can cause mutation? Any idea how can mutation happen? There are many types of mutation which is not in your syllabus, like single gene, we call it base pair, single base mutation, and insertional mutation. Or so single base mutation is you have a codon, right? A, T, G, C, A, T, let's say. And this particular A got converted to a, let's say, G. And the code now becomes ATGCGT. This is a mutated sequence. Do you understand? This is called a single base mutation. Is it clear?
Yes, sir.
This happens in sickle cell anemia. Write down. You know sickle cell anemia? What is this?
Shape of the
RBC
RBC changes.
That happens due to a single base mutation. This is first type, single base mutation. Let's not do factors right now. So you understand single base mutation when one single base is mutated or changed. The other possibility is insertional mutation. So there is a normal sequence, again, ATGT, AGC, let's say. And at this locus, between G and T, there is an insertion that happened, because of which the sequence become ATG, A, TAGC. Can you see this? This A got inserted here, because of which earlier when you were reading the codon, remember I told you there is 3 bases we read at one time. It's called a codon. So this was the codon, right? Now it changes. Now it becomes this ATG, which is correct. TAG, it's become ATA, GCA. You understand? So due to insertion, same can happen, which is like deletion mutation, insertional or deletions. One base can get removed or left out or deleted. In both this mutation, let's also do this. Let's say ATGT, ACT, AG. Then if this C gets deleted, then it will become ATGT ATAG, right? Earlier the code was ATG, TAC, and TAG. Now it becomes ATG, TAT, AG. So because of insertional or deletional mutations, you can write down the reading frame. This is not in NCERT, but can be asked. But this part is there, that insertional mutation, deletional mutation, and single base mutations are there. So reading frame of the gene or of the DNA changes. Okay. Due to a single base mutation, it happens only at one base. So earlier it was ATG, CAT. It will still be ATG, but CGT, and everything remains the same. Is this clear? Making sense to you, everyone?
Yes, sir.
Yeah, very good. So this kind of mutation where this shift, the reading frame changes, it's called, both of these types are known as, just remember this is in NCERT, so it can be asked in the entrance. They are called frame shift mutations. Okay. Both insertional and deletion mutations. Done? Please note down. Let me know when you're done. Done, people? Saifullah, shall I move forward?
Yes, sir.
Yeah, done? Okay. Good. Now let's come to factors now. What do you think can cause mutation? So whatever causes mutations are known as mutagens. Okay. Okay. So the agent, write down, agent causing mutation is known as mutagen. Now, mutagen can be a physical factor. For example, what are the examples of physical mutagens? Any idea? UV radiation. Remember I talked about UV radiation? It is a very high energy radiation. Everyone? It is a physical factor which causes mutation. Okay. Or it can be chemical factors. Right? What is a chemical factor that can cause mutation? Any idea? Am I audible, everyone?
Yes, sir.
Yes. So any idea what is a chemical mutagen? Have you heard of this? There's something called nitrosourea. Okay. Write down. Nitrosourea is a chemical mutagen which can cause mutations. Is it clear?
Yes, sir.
Mutations, anyways, like there is a mutational load, but I think that's very out of your syllabus. So let's not discuss that. It will not be asked. If you pursue higher education in biology, then you will study about more mutagens and the process of mutagenesis, how basically it happens in each and every cell. And cell tries to repair it also, cell tries to get rid of it also. It's not like cell is okay with any mutation. Because see, if a mutation happens in a very lethal, very vital gene, let's say there is a gene which is important for metabolism. And if that gets mutated, the organism will just die, right? Yes. So any living system will not very easily just accept mutations, but I think this is all you need to know for your entrance and board's point of view about mutation. Yeah. Yes. Okay. So now let's come to the final, but a major chunk. I will start it, probably not finish it today. To study genetics disorders, study of genetic disorders. We discussed in chapter 4, where we discussed medical termination of pregnancy, right? We did discuss some genetic disorders. Remember, everyone? So before we study genetic disorder, we have to understand how do we conduct this study. Now, to study genetic disorder, there is one thing that we all should know first, is called pedigree analysis. You know what is pedigree? Yes, Minashi. You know what is pedigree analysis? What is pedigree basically? Any idea? These are symbolic analysis which we do where let's write down. Pedigree analysis is the study of family history. Pedigree analysis is the study of family history about inheritance of traits. About inheritance of traits, disorders, or diseases, anything, okay, which is genetic. Traits, disorder, or diseases through symbolic representation of individuals. Okay, that's called pedigree analysis. Is it clear, everyone?
Yes, sir.
Very well. Now there are certain symbols that you have to study in a pedigree analysis. This is the standard notation that if there is a square, and be very careful when you are making it. If it is a square, it means male. Okay. And if it is a circle, we are denoting that with female. Now this is very interesting when we do this, some exercises on this. So square is male, circle is female. And if for some disorder, the sex is not specified or you do not know about the sex of that individual, but you know that there was someone who carried or transmitted that gene or disorder, but you don't know whether it was male or female, then we use this. What will you call it? Diamond? Shall I call it diamond? Diamond or parallelogram, whatever you think. It is used for sex which unspecified. Okay. When we do not know about the sex. Is it clear? Now, most of the papers, most of the questions, all these three, if they are affected, like if they are carrying a disease, they are colored. Like they should be red colored, but if it is a black and white paper, they make it black colored also. But just you should know, if it is a colored square, a colored circle, or a colored diamond, what does these three states means? It can just write down colored. This means affected or diseased. Diseased individuals. Is it clear? And if it is just blank, just an outline and no color is there inside, then it is just unaffected, male, female, or sex unspecified. Okay. Now, if there is this kind of a notation, there is a square connected to a circle. What does this mean? This shows mating or marriage. Okay. So this male is married to this female or mated to this female. Is it clear?
Yes, sir.
And if it is shown by double bonds, then what does this mean? This is again mating or marriage, but this time the difference is the double line is showing a greater connection or bond, so it is mating or marriage between relatives. Between relatives who are either blood relatives or are like somewhere connected, like cousin, first cousin, second cousin, but comes in that family only. So this is and this has another word, it's called consanguous marriage. Consanguous, N-E-O-S. So marriage in close relatives is shown by a double bond. Is it clear, everyone? Now, using this, let's try to make a small pedigree. So if I'm making it like this, I showed it like this, and then there is a line coming and it shows this. What does this mean? What does this pedigree mean to you?
A couple has a daughter and a son.
Yes, so this means these are the parents and these are the offsprings. Okay. So, and this means, you're right, parents are above. When parents or couple, as Jasola said, above, produced offsprings which are below, and how do we, if I tell you which one was born first, this girl or this boy? Can you know how to figure that out? So the order of birth is from this direction, from left to right. Okay, write down. Order of birth is from left to right. So can you tell me which is elder? Who's elder, the sister or the brother?
Sister.
Yes, so it has a elder sister. From left to right is the order of birth. Very good. Let me ask you one more thing. If I make a pedigree like this, so I'm making this, and then this is colored. What does this mean? What does this mean? And it
They
also
have
has
a
a,
deceased son.
yes, and so this means that parents who were not diseased, having a affected or diseased, whatever disease we are studying, they have a diseased son along with a and a normal daughter. Yes. And who's elder? The son. Okay, everyone. Sometimes there is one more notation that is used, which is numbered. So if they make it this and number it, let's say 4, what does this mean? This simply means 4 unaffected. This is not used much, but offsprings, because the sex is not specified. Can you see? Yes. So this means 4 unaffected, unidentified offsprings. And if the same is done like this, and it is written 4, then what does this mean?
4 male.
4 unaffected males, right? And if it is this and there is written 4, then same. 4 unaffected females. Is it clear? All these pedigree analysis symbols are clear to you? Everyone has noted everything?
Yes.
Yeah. Okay. Now, what we have to study is, how much time do we have left? We have some time. I can start with one concept and can finish it. It should not be left in between. So, understanding the pedigree analysis, there are certain types of diseases, genetic disorders are of certain types. Just like genes are of different types, they can be dominant genes, they can be autosomal genes, they can be sex-linked genes, or they can be recessive genes, right? So if I say, let's start with first concept. If I say there is something called autosomal dominant disorder. So can you tell me, Saifullah, what does this word tells you? Autosomal dominant disorder. What does it tell you?
Non-sex chromosomes related.
So autosomal means it is related to?
Autosomes.
Yes. Through autosomes. Or the gene is present in autosome, which means it is non-sex. So it is not linked to any sex chromosome, correct? And what does the dominant word tells you? Dominant. It can express in a heterozygous state, right? Yes or no?
Yes.
Because it is a dominant one. So even if one allele is present, it can be expressed. Versus if we just go to the second part, which is autosomal recessive trait. How do you think it will be different from autosomal dominant? Again, not linked to sex, right? Correct? And this time it is recessive. So what does this mean?
Express in homozygous.
Yes. Expresses in homozygous state. Correct? Now, let's write down some examples that are asked in entrances. So for this autosomal dominant, the one that we will be also studying is dystrophy. It's called muscular dystrophy or myotonic dystrophy. The word myo is for biology terms. The word myo, muscle. So myotonic dystrophy or muscular dystrophy. And autosomal recessive is what we studied above, sickle cell anemia. Sickle cell anemia. Now, I am going to give you a pedigree. Okay, everyone? You have to analyze it as a, this will be the exercise that we'll do. And you have to tell me whether it is an autosomal dominant disorder or an autosomal recessive disorder, whether it belongs to myotonic dystrophy, it's representing this or that. Okay. So let me draw it. Let's say a female get married to a male and they had, let's say, 4 kids, girl, boy, another boy, and another girl. And it's like this. Mhm. Okay. And the affected ones are okay. So this is the complete pedigree analysis. You have to tell me, you can number or use alphabets, and you have to tell me the analysis for this pedigree, and also tell me whether it denotes autosomal recessive or autosomal dominant condition. You have to give me reason why do you think so. Okay, everyone. Recessive is the first question. Second, why? Justify. Okay, Saifullah, Mamuna, and Minashi. You see this pedigree?
Yes, sir.
Yeah, tell me. Do the analysis and tell me. Take 2-3 minutes. I'll be back. Okay, I'll get myself some water. Okay, Saifullah.
Okay, sir.
Okay. Hello, everyone. I'm back. Am I audible? Invisible?
Yes, sir.
Done? Solved?
Yes, sir.
Yes. So Saifullah will help me. I'll follow the lead. Saifullah, what do you think? Which one is it? Is it autosomal dominant or autosomal recessive?
I think it is recessive.
You think it is autosomal recessive. Okay. So, and why do you think so? Others, let me take others' answers first. Minashi, what do you think it is?
Autosomal dominant.
Autosomal dominant. So you think it's dominant. Mamuna, what do you think?
Sir, for me also autosomal dominant.
Dominant. Okay. So let's go to Saifullah first. Saifullah, why do you think it is recessive?
So it is mostly being expressed in homozygous condition.
Okay. And is it really? Mamuna, why do you think it is dominant?
Because there in the first case the female is affected, so it just transfers to the son, and the second child doesn't get transferred any the disease, but the third child gets transferred. So I think it's dominant because it's been transferred to majority of them.
So this is a female or a male?
Female, right?
Yes, it is a female. So if you think it is autosomal dominant, let's start it like this. So it will have 2 chromosomes, right? On one chromosome, this disease is present. So I will write D, because it's, if you think it's autosomal dominant, only one copy will be enough, right?
Yes.
And even on the other strand, it is nothing. Then, and for a male to be normal, the male needs to be negative for both, right? Because if there was any on any one of it, it would have, it cannot be a carrier. So either she is diseased, she is normal, he is normal. The first kid, this is a male who got affected. Now, if the father is normal and the mother is affected, and a male got disease, it clearly tells that it got this disease gene from the mother and the normal gene from the father, right? This one, do you understand, everyone?
Yes, sir.
So it clearly, you don't have to go to others even. It clearly tells that this female got a normal from mother and normal from father. This male got a normal from mother and a normal from father. This female got a diseased from mother and normal from father. So it is happening in both the sexes, and it is happening with a normal father. So clearly, only this, studying this tells us that this is autosomal dominant, right?
Dominant.
Do you understand why it is autosomal dominant now, Saifullah?
Yes, sir.
Then again, this mother was diseased and this father was negative for both. And this here received diseased gene from the mother and normal from father and got diseased. These two received normal from both parents and got nothing, right? Do you understand why? First, because it's happening in the next generation with a normal father who cannot be carrier. You cannot be a carrier for autosomal dominant. Can you be a carrier for autosomal dominant? No, because any dominant trait can express itself even if one copy is present. Do you understand, everyone? Yes or no? Saifullah, yes or no?
Yes, sir.
So write down, autosomal dominant diseases can express with even one copy, one disease copy. So the father in this case is normal. And the only way then for the son to get the disease is because that disease is autosomal dominant and can travel in the next generation with just one allele faulty. So this represents myotonic dystrophy, right? You understand, everyone? Not sickle cell anemia. This is correct. This is wrong. Everyone understands?
Yes, sir.
Pakka. In the next class, which is tomorrow after your test, we will be taking more examples of genetic disorders from both autosomal dominant and autosomal recessive. And then we'll go to the sex-linked dominant and sex-linked recessive. Okay. And in the end, we will go to chromosomal disorders, which are basically the which we have already discussed a little bit. Trisomy of chromosome 13, 21, right? Down syndrome, Tau syndrome, Turner's, Klinefelter's. We will do that, and that will end the chapter. So tomorrow we'll aim to finish the chapter so that we can start with the 6th chapter. And when we'll finish the chapter, before starting the 6th chapter, we will do NEET based questions on this chapter and a lot of pedigree analysis based questions as well, which has come in previous years exams. Is it clear, everyone?
Yes, sir.
So please revise, and all the best for tomorrow. And I'll be there. and ask me if you have any doubts tomorrow.